Welcome to part two of our discussion of genetics and inheritance. In the previous lecture, we talked about some basic terms of genetics. And in this lecture, we'll be applying some of those basic terms and also looking at things with Punnett squares and pedigrees, and we'll be applying it to actual patterns of inheritance and seeing how that plays out in how it relates to specific diseases and then how those diseases are then passed on from generation to generation. So our objectives for this part would be to know the differences between autosomal and sex chromosomes, which really does play into the patterns of inheritance, which leads to point 2 or objective 2, in which you should be able to describe and be able to identify patterns of inheritance. Finally, I'd like you to be able to explain X inactivation and discuss mitochondrial inheritance, another pattern of inheritance that we look at the very end of this lecture. So to begin, I want to define the terms of autosomal or sex length, or the difference between autosomal chromosomes and sex chromosomes. So in a normal individual, you have 23 pairs of chromosomes, or chromosome pairs, and these are homologous chromosomes. As you can see here in our karyotype, which I have discussed with you in previous lectures, is an image that shows all the different chromosomal pairs, the 23, and how they break down. It's a way for geneticists to look at the health of these chromosomes or how they are structured. Now this karyotype, or the chromosomes within an individual, break down to two types. We have the autosomal and the sex chromosomes. So the autosomal chromosomes are the 22 non-sex chromosomal pairs. So they are labeled 1 through 22 in our karyotype here. And then we have this pair at the end. These are the sex chromosomes, the either X or Y. If an individual is XY, he is male. If an individual is XX, she is female. So those X or those sex chromosomes are the gender determining chromosomes. Now, when we talk about inheritance patterns, the association of genes located or at the locus is on either autosomal or sex chromosomes does play out in how that pattern propagates. And we'll talk about this in great detail in the remainder of this lecture. So I want to begin talking about autosomal inheritance patterns, and the first one we'll talk about is autosomal dominant. So when we talked about dominant and recessive previously, we talked about how a dominant trait masks the recessive trait, in that if you have one allele that is carrying the dominant trait, it will mask the recessive allele. So if you have one, you will have that. And when we're talking about disease, and that's kind of been our main focus for traits within this lecture, is we'll talk about how if the individual is affected or not, or be affected by that disease trait. So in an autosomal dominant pattern, affected children usually have affected parents. So if we're looking at this individual here in the star indicated right here, this individual is heterozygous in that it has one dominant and one recessive allele, but the the dominant allele is carrying a disease state. So all heterozygotes within this family or this pedigree are affected, or anybody carrying this dominant allele would be affected. If we look at the parents of this individual, these parents were both heterozygous in this case. They were able to have the one child with heterozygous carrying the one dominant trait and one recessive. There was also one affected individual here. You can see they don't know the other allele, but it doesn't matter because it's carrying the pattern is carrying the dominant allele, so that individual is being affected. The other two siblings you can see here are homozygous recessive, in that they are not affected and therefore they do not carry that gene. So these individuals then in subsequent generations do not pass this gene on to their offspring. So this subsection of the family is not affected in the autosomal dominant pattern. Two unaffected parents will usually not have two unaffected parents, as we said here, will not usually have unaffected fields. And as you will notice, the males and females in this pattern are equally affected, in that you can see it's 3 males and 3 females are affected. There's no clear indication that it's clearly with the males or strongly with the females. So as you can see here, this is a good example of the autodominant fashion of inheritance, in that the autosomal pattern is most often affected. If you're heterozygous, you are affected by this disease. It goes through multi-generations, and it equally affects males and females. So in this slide is another way we can look at the autodominant pattern, in that we can kind of bring in the Punnett squares, which we talked about in the previous lecture. So here's our pedigree at the top, where you have the affected individual carrying the dominant allele here, the uppercase A, mated with one normal parent carrying both recessive alleles, or unaffected. They have a 50% chance of showing individuals that are affected and a 50% chance of showing individuals that are unaffected. So how does this break down with the Punnett square? So as I mentioned before, the one parent has as the dominant allele and a recessive allele, so he's heterozygous, and then the normal parent or unaffected parent has 2 recessive alleles. When we use these alleles to then fill up the Punnett square, you can see here that the 2 individuals on the left column here have are heterozygous, carrying the dominant allele and the recessive allele, while the individuals on the right side only carry the recessive allele. So if we break this out, and it's really about probability, there is a 50% chance that the individuals deriving from this mating of one affected parent that's heterozygous and one normal parent is a 50-50 chance that they will either carry, that they will be affected or not, and there'll be 50-50% chance that they will also be carrying affected and carrying the disease trait. Now, if we mated 2 affected parents, so they were both heterozygous, one carrying both heterozygous, so we have the dominant and recessive allele, and then we play this out in looking at the Punnett square. So we had our heterozygous with the dominant and recessive, heterozygous with dominant and recessive. We bring them filling out the Punnett squares as we do. We can see that this one individual is homozygous for the dominant trait, therefore he is affected. Or 25% are homozygous for the dominant trait. 50% are affected and are heterozygous, carrying the dominant and recessive trait. And then the remaining 25% of the offspring are unaffected and are homozygous recessive, in that they are not being affected by this. And this plays out once again as well when you look at the pedigree. Now, these offspring possibilities or probabilities are generated if you go through this breeding independent assortment through multiple generations, but rounding out, the main thing is that these are the general probabilities if this mating or breeding was taking place in multiple times over and over again. So now, as with each of these inheritance patterns, I kind of wanted to bring in an example just to have something to kind of concretely tie into, or something you may have observed in your practice, just giving examples of these inheritance patterns. So one example of autosomal dominance is post-axial polydactyly. And polydactyly means extra digits, so you have a presence of an extra digit next to the 5th digit, which means you have an extra finger next to your pinky finger, as demonstrated by this individual here. Now, this follows the autosomal dominant pattern in that it equally affects both sexes. Transmission happens from both sexes. It doesn't matter. You can see it's passed on from generation to generation, and you are observing it in every generation, right? If it's dominant, there's a very good chance that it will be passed on. So you can see that we have this multi-generational pattern in this autosomal dominant pedigree here. One more example is Marfan syndrome. This is an autosomal dominant pattern disease. It is somewhat common in that it's in 1 in every 5,000 births. It is a connective tissue disorder in that you have very essentially these individuals will become very long-limbed, or they're tall and lanky. You have some issues with the stretchiness of these disease-connected tissues, very important for them. Holding these together and allowing for elasticity and stretching. So these individuals are very tall and lanky. They have very long, flat feet and long fingers. Also, a large aortic root. A big risk of health for these individuals is instances of overexerting themselves or being too much exercise. The aortic root can burst due to not being able to respond to the stress in a high exercise state. So these individuals, while you would think they would be prone to be good basketball players, stereotypically due to their size, have a very strong risk and potential danger of being these high exercise states, in that they could have this aortic rupture. So it's a common thing that they look out for in individuals that are demonstrating these symptoms. Some examples of Abraham Lincoln was thought to have Marfan syndrome. It wasn't completely diagnosed, but they looked at that. King Tut was also potentially an individual they thought that may have Marfan syndrome. So these just kind of some examples from history just relating Marfan syndrome. So now I want to move on to autosomal recessive inheritance pattern. So this will be a little different in that we're dealing with the recessive and not the dominant. All right, so that means that individuals with the recessive allele would be affected, but only individuals that have both alleles that are they have to be homozygous for the recessive allele. The dominant allele would be preventing them from being observed in, or preventing them from presenting the disease state or being affected. So you're only being affected if you are homozygous recessive. All right. Now, in this pattern, both males and females are equally affected. So in the autosomal inheritance patterns, the sex doesn't play into it. It's not directly linked to sex. When we get to sex, that will change, but we'll get to that a little bit later. But in all these autosomal patterns, males and females are equally affected. Most parents actually who are affected in an autosomal recessive do not pass the affected state on to their children. The children do carry the allele, because when you cross this parent who has the homozygous recessive allele, obviously it has to give one allele to their offspring. So these offspring are often heterozygous, but heterozygous carrying the disease and unaffected allele. Now, these diseases are much less common because it's very difficult often to get the both recessive alleles, so they're less common than we are observing in the dominance pattern. But what can happen, as you can see down here, in that in this pedigree, we had the individual affected female. She had two females who were heterozygous recessive, but they weren't showing, or they weren't affected by the disease. And then what happens here is you had cousins breeding. So this is why interbreeding is often bad, because you have the arisal or appearance of these autosomal recessive diseases arising. But when this was breeding, so you had heterozygous recessive, or heterozygous showing the recessive trait here, heterozygous showing the recessive trait here, this allowed for these two recessive traits to arise and having offspring from this cousin breeding pattern to allow to have these two individuals, male and female, showing the affected trait because they are homozygous recessive. So this is once again another good reason why interbreeding among very close relatives is a problem. And oftentimes, many of these autosomal recessive traits are commonly presented in specific family lines or regions where people are trying to maintain some sort of, not necessarily interbreeding, but you're selecting your mating partners from a small population, which can eventually lead to recessive traits being able to arise and showing disease states. So now, as we demonstrated with the autosomal dominant, I wanted to kind of walk through and relate these pedigrees to the Punnett squares to kind of give everybody just another way of looking at this inheritance pattern. And it also kind of shows a good idea of the probability of the offspring being affected. So we'll start on the left side here. When we have a heterozygous carrier parent breeding with one affected parent, so the affected parent will show both recessive traits or recessive alleles, while the unaffected carrier parent only has one recessive allele. So when we fill that out to the Punnett square, we can see here that 50% of the offspring have the probability of being affected in that they're homozygous recessive. The other 50% are going to be heterozygous in that they carry one dominant and recessive allele. They are not affected, but have the chance, they will pass on most likely that recessive allele and could have the chance of having affected offspring if they are mating with individuals that also carry the recessive allele. This leads into the example on the right side where two heterozygous carrier parents both carry the dominant and recessive allele breed, and you can see here this leads to this resulting Punnett square as demonstrated down here. Now you'd have 25% of the population would not carry the recessive allele, so they would not be affected and would not be able to pass on the recessive allele to their offspring. 50% would be heterozygous in that they are unaffected but still pass on the gene to the affected gene to the next generation. And then you have 25% of the population that is homozygous recessive in that they are affected and carry both recessive alleles, so they will pass on a recessive allele to their next generation. An example of an autosomal recessive disease is cystic fibrosis. It is somewhat common in that it's affecting 30,000 individuals in the US. It is a mutation in the cystic fibrosis transmembrane receptor, which you can see here demonstrated in this image. It is an ion channel allowing for movement of things across the plasma membrane. What ends up happening is this defective ion channel leads to a mucus buildup within the lungs, causing difficulty breathing for these individuals. And you can see this gentleman down here is using a nebulizer and a percussion vest to loosen up that thick mucus. The nebulizer helps thin out the mucus, and the percussion vest, that physical vibration, helps loosen things up to help these individuals breathe better. So cystic fibrosis is an example of one of these autosomal recessive diseases. So we have completed our discussion of the autosomal inheritance patterns, and now I'd like to move on to the sex-linked inheritance patterns. And the sex-linked inheritance patterns are talking about genes that are located on the sex chromosomes, which leads to changes in this pattern. It leads to significant alterations in that there is very clear male to female increases of chances or decreases of chances depending on if it's on the X or Y chromosome. So we'll look at these patterns. We'll look at X-linked recessive and X-linked dominant. We also looked at Y-linked. And the X-linked, you should note and remember, is interesting because females have two X chromosomes, while a male only has one X chromosome. So this leads to interesting patterns that we'll observe in the upcoming slides. So we'll start off with X-linked recessive. So what this means is that the gene is located, or the affected gene is located on the X chromosome. Now, as I mentioned before, females have two X chromosomes, while males only have one. Now, as we know when we talk about dominant and recessive genes, the recessive genes are not showing the affected state, or they're not presenting an affected individual when there is a dominant gene around. However, in males, when there's only one, you can express that recessive trait or disease state because there is not a dominant trait to even have a chance to masquerade. It's whatever you have. So in this individual here, we have a grandfather who is carrying the recessive recessive gene here on the X chromosome. Now this grandfather and grandmother had offspring, leading to a daughter and a son. The daughter is carrying a dominant and recessive allele, but the recessive allele is being masked by the dominant allele on the X chromosome. The son did not get the son could not get the X chromosome from his father, right? Because you needed to get one chromosome from the mother and one sex chromosome from the mother and one sex chromosome from the father. So that son got one X chromosome carrying a dominant allele from the mother, and then it got the Y chromosome from the father. All right. So this son did not get the affected or recessive allele from the father, but if that son then mated with a female who had two recessive alleles on the X chromosome, when they were in their resulting offspring, all the sons would be affected because the Y chromosome was always passed from father to son. The son would have to get an X chromosome from the mother, and so the mother, in this state, because she had both recessive alleles, would have to pass on a recessive allele to that son. So it would lead to a disease state from that. Now, the other interesting thing about X-linked recessive genes is that they can skip generations. So it often goes from grandfather to a carrier daughter to a grandson. So it would go from grandfather to grandson. So remember we talked about here, this daughter would be an unaffected carrier in that she is heterozygous for this gene on the X chromosome. So you can see the recessive trait here, the lowercase b. This daughter then mated with an individual who was carrying a dominant B gene, you can see here. Now, we had a son and two daughters who were unaffected, but this grandson, the grandson from the affected, or the son from the affected daughter, got a Y chromosome from the father, and then it had a 50% chance of getting a dominant or recessive allele from the daughter or his mother. So he had a 50% chance, and it was presented with the recessive allele, so this would be an affected grandson. And so you have this pattern of skipping from one generation from the grandfather to the grandson being affected because you had the unaffected carrier daughter. Now, if we look at this once again in our Punnett squares, we can see how this plays out. So if you have an affected father, and you can look at the chances it has of passing on. So this affected father had on the X chromosome an affected gene, and it can only contribute in the sex chromosomes either X and Y, and then the mother unaffected carrying on the X and Y, so passing it on. So an affected father cannot pass on the affective gene to his son because it's not on the Y chromosome, it's on the X. The father can only give a Y chromosome to his sons by definition. The mother can only giving X chromosomes, and so the mother is giving an X chromosome to the daughter, and to the daughters in these situations. Now the father can only give an X chromosome to his daughters by definition as well, because daughters are XX, so it'd have to get one chromosome from the mother and one chromosome from the father. And the father only has an affected or X chromosome carrying the recessive allele. So therefore all the daughters would be carriers because they would have an affected and unaffected allele, but since it's recessive, it's being masked, so they wouldn't demonstrate or show the state. They would disease state, they'd just be carriers. The sons, as we previously talked, were unaffected because they can only get that Y chromosome from the father. If you had a carrier mother and an unaffected father, as shown in the pattern on the right here, so the father is unaffected, contributing an unaffected X and an unaffected Y to all his offspring. In the chance of the sons, there is a 50% chance the son would be affected because the father, by definition, has to give the Y chromosome, so it has no contribution. But you have to fill out with an X, so it's either 50% chance that the sons would either get an affected X chromosome or an unaffected X chromosome, as you can see here. So there's a 50% chance the son are affected. This plays out similarly with the daughters in that the daughters would never be affected because they have that dominant allele masking it from the father, but 50% of the daughters would be carriers as demonstrated by the upper left quadrant of this Punnett square here. So an example of an X-linked recessive gene is hemophilia. It affects 20,000 individuals within the United States, and it is a blood clotting disorder in that individuals with hemophilia have a very difficult time clotting blood if they are cut or if they proceed to bleed. The blood does not coagulate, it doesn't clot properly, and doesn't stop bleeding, so they continue to bleed. So it's very dangerous to these individuals in that they could, once they start bleeding, they have a very difficult time stopping. There are treatments where you can supplement the missing coagulation factor. In a later lecture, you'll talk about hemophilia, a little more details discussing these missing coagulation factors and how this pathway plays out. But hemophilia is a good example of this. I personally actually have hemophilia within my family. My great grandmother was a carrier. She was not effective, obviously, as most females are not. She passed this on to my great uncle. Two of my great uncles were affected by hemophilia. My grandfather did not get an X chromosome carrying the hemophilia gene, and so he was unable to pass it on down to me. So my family, my mother was not a carrier, and nobody in my family from my grandfather is being affected. But I have other relatives, my great uncles were affected by hemophilia, and so it is very common, and it does hit me home personally. And I always find it interesting looking at, and it got me interested in looking at pedigrees and Punnett squares in looking at my family history with hemophilia. Hopefully none of you are affected by such these diseases. However, if you are, it is kind of an interesting look at your family tree and family history and looking at how these patterns of inheritance are passed down. So we talked about X-linked recessive. Now I'm going to move on to X-linked dominant. So X-linked dominant, obviously, you're going to then start pulling in more of the females to be actually effective, right? Because that dominant allele is masking a recessive allele. If you have that allele that's dominant, you will be effective. So therefore, females are 2 times more likely to be affected. Moreover, the father to son transmission is not observed, and all daughters are effective if the father does have it. So let's go back to the father-son transmission, right? If the father is affected, the father carries obviously an effective X chromosome, but the father is going to his sons will only give the Y chromosome. So it is not passed on from the father to son. You don't see that pattern. As you can see here down with individual 7, who's to be effective father, he has no sons that are effective, only his daughters are effective. Now, all daughters are effective from the father because all daughters would get an X chromosomes for the father, and if the father is affected, then it has to pass on that X chromosome with the affected gene. So all daughters of that father would be effective, as you can see here. Now, effective females usually are heterozygous, and they have a 50% chance of passing on the disease allele to the daughters or sons because one X chromosomes would be affected, 1 X chromosomes would be not, and so you have a 50-50 chance if it passes on to son or daughter, and that's an equal chance because the mother always gives an X chromosome to either the son or daughter. Now, an example of this X-linked dominant pattern would be generalized hypertrichosis, which is better known as werewolf syndrome. As you can see this individual over here, it is a very high excessive hair growth growing in abnormal areas. And so you can have these individuals that become extremely hairy. It's very noticeable in the face. They have hair growing all over the place in an excessive pattern. Now, this is a generally rare condition affecting less than 50 individuals worldwide. And in general, these X-linked dominants seem to be a little less common than you are seeing in the recessive patterns, as demonstrated by the higher prevalence of hemophilia. Now, before we move on to Y-linked, I just wanted to touch on one concept involving X chromosomes that I really wanted to bring up, and I thought it would be important when thinking about these inheritance patterns and what happens with them. So the term is called X inactivation. So as you know, females have 2 X chromosomes and males have an X and a Y chromosome. Now, this chromosome contains genes that has functions, and there's a lot of important cellular functions that are located on the X chromosome. Now, it's important to maintain balance and proper health or genetic health or proper protein expression, that we need to balance out between the males and females the number of X chromosomes or the genes being expressed on the X chromosome. And so what the body does is it inactivates one of the X chromosomes. It forms what's called a Barr body, which is a very highly condensed form of one of the X chromosomes. And so we talked about previously, condensing of chromosomes inactivates them. So what happens is the body will then inactivate, as you can see here, or very highly condense one of these X chromosomes into a very small state. So that is not being used to transcribe new genes. And so it does this to kind of balance out the normal function of the cell, to maintain normal balance within the body. What is interesting is that this concept of mosaicism, in that which chromosome is being inactivated, can vary by cell. And so you can have mosaicism would be like a mosaic, where you have different pieces of things being fit together to build a larger picture. So in the body, what you could have is you could have slightly different genomes, in that you have a pattern of expression of these cells, that some express one X chromosome, some express Y, and when are this inactivation occurred and which one occurs can kind of determine if there's certain parts of the individual that have more of one X chromosome being present, or another X chromosome being present. So you get this mixture, this regional mixture of certain cells or tissues even, that are expressing and using one X chromosome, while there could be another one that's using another X chromosome. An example of this would be like there's these cats, right? There's a medial line down the middle where they're either black on one side and then brown on the other. This is an example of X inactivation, and one side is expressing on that chromosome for the cat, showing that it expresses the brown phenotype, while on the other side of the cat it would be expressing the black phenotype. And it's really just the inactivation of one of their chromosomes linked to that. So that's just a good example of how this mosaicism works. And this does happen within humans in a similar state, in that you will have certain, in females particularly, there obviously with the 2X chromosomes, there will be some parts that will express an affected state while others would not. And this does lead to some of the variation in the expressivity or the severity of disease within individuals with carrying potential disease states or diseased alleles. So now I'd like to move on to Y-linked inheritance patterns. We talked previously about all the X-links, dominant or recessive. Now Y-linked, there's only one Y chromosome. So we don't deal with dominant or recessive. It's just if it's there or it's not. Now Y-linked is a smaller, less significant, we'll say, inheritance pattern, in that the Y chromosome contains less than 300 genes. And so there's less diseases really physically linked to the Y-linked chromosome. As you'd expect in a Y chromosome being passed on, it goes from father to son. It does not affect any of the females because they don't carry a Y chromosome. And so you get this Y-linked pattern going from father to son to sons, or father to son to son. Know that none of the parent females are affected in a Y-linked inheritance pattern. To finish up our discussion about sex-linked inheritance patterns, I wanted to kind of finish up with a distinction between sex-limited and sex-influenced traits. So sex-limited traits are traits that are only occurs in one sex. The best example of this would be the clear anatomical differences between males and females. And so this is an example of sex-limited where you're just specifically looking at one trait. Sex-influenced are interesting in that it's a more common in one sex, but it can occur in both sexes. So an example of this would be hair loss. So as you can see here, an example here, this male is showing the standard male hair loss pattern, losing somewhat up in the front and then thinning out in the back as well, and eventually leading to full hair loss in the typical standard male pattern baldness pattern. This is associated as an autosomal dominant trait that is very prevalent in males, and you can see it being passed on in a very similar fashion, in that most males are passing on this male pattern baldness, or at least a high percentage are. Females' hair loss is an autosomal recessive trait in that females express a different pattern, but they do also experience hair loss. So this means that the sex is influencing how this trait is being passed on and how it is being presented. So you have that sex influence aspect of this. So the difference between sex limited, where you're talking about the anatomical differences, where it's only happening in one sex, versus sex influence, where it can occur in both sexes, but the pattern or severity is affected by the sex of that individual. To finish up our discussion of inheritance patterns, I wanted to talk about mitochondrial inheritance. So mitochondrial inheritance is another way that genes or affected states in the diseases can be passed on. So mitochondrial are kind of mini cells within our cells. They are the powerhouse of the cell. They have their own genome, and so if they have their own DNA, they can pass on that to the next generation. It can be passed on. But the interesting thing is that the mitochondria are passed on from generation to generation in the oocyte, or the egg from the female. And so you're only getting transmission via the maternal line or by way of the mother. So those mitochondria go from mother to daughter and son. In the males, the sperm doesn't carry any mitochondria with it, so it cannot pass on that to the next generation. Now, due to the fact that the mitochondria within the female, there's lots of them being passed on, can lead to lots of different variations of genes because there's just packed with mitochondria, and so you do get a large degree of phenotypic variability because you're not clonal, they're not all the same. They have larger variants, which does lead to variance in the phenotypic probability, the expressivity, and also the penetrance, which we will talk a little bit about in the next few slides. So an example of this inheritance pattern, as you can see here, you can see the mitochondrial DNA are being passed on from the mother to son or daughter. Now this son, obviously the sperm doesn't pass on the mitochondria, so the son's family is unaffected. However, the mother passing on to the daughter then can pass it on to the next generation. Once again, though, that son of that resulting daughter can't pass it on because males can't pass it on, but the females will once again be able to pass it on to the sons and daughters as well. So it only passes on through the mother in the example of these mitochondrial diseases. And an example of a mitochondrial disease is Leber hereditary optic neuropathology, or neuropathy, or LHON. This is a rapid loss of vision due to optic nerve death. which starts in around in the 30s or mid 30s. And so you can see here's an example of this from the Eye Society, where you have an unaffected individual. You can vision appears like this. But in LHON, you can see that you start to get blurring of the image and loss of vision in the center of vision, in that you're starting to die, that optic nerve is starting to die, leading to loss of vision in this pattern. Now, this is a missense mutation in the protein coding gene, leading to a large change in the protein coding gene, eventually leading to the disease state and loss of vision. This is once again an example of the mitochondrial inheritance pattern. Finally, I just wanted to wrap up, as I always do, reviewing our objectives. So you should be able to know the difference between autosomal and sex chromosomes. So autosomal will be those 22 chromosomes, while the sex chromosomes are the remaining pair, the X and Y chromosome. We talked about the different patterns of inheritance. So we talked about autosomal dominant, autosomal recessive. We talked about X-linked dominant, X-linked recessive. And we also talked about Y-linked, even though they're very rare and not really hugely significant in the overall population. We briefly discussed X-linked inactivation, or X inactivation, excuse me. And then finally, we finished up talking about mitochondrial inheritance, how things are passed on in the mitochondria from the mother to their offspring. I hope you enjoyed the lecture, and I will talk with you once again in part 3.